see yourself back there.
Where? Where would I see myself back? Okay. Okay.
Ready to go?
I think so.
Okay.
Yeah.
Okay. Well, welcome. It's my pleasure to introduce Dr. Renzo Shamey, who I am learning loads about color in the textiles world, and he has been gracious enough to even attend my class, and I can talk to him about digital imaging on occasion. Turns out that Dr. Shamey and I have several things in common there. We both spent a fair amount of time in the UK. He got his degree there at Leeds, very famous place for textiles and color. But he also then spent a year at Harriet Watt University, where I had spent a sabbatical way back when there. So we can talk about Scotland together. I note that when he was at Harriet Watt, he won an Excellence in Teaching Award. So we are in the presence of an excellent teacher, which is always good. He joined the textiles. I love it. It is textiles, engineering, chemistry, and science at North Carolina State University, and in 2003. So relatively new here. And I am tickled pink to hear the gentleman talk.
All right. Well, good afternoon, and thank you very much for that introduction. As Dr. Trussell said, my name is Renzo Shamey, and I'm the Director of Polymer and Color Chemistry program in the College of Textiles, and we have an undergraduate program that has color science as a core component. We also have a graduate degree and a number of graduate students, PhD students, that are working on color-related problems. So the title of presentation today is Perceived and Predicted Color Differences, and the idea is hopefully to introduce some of the issues that relate to the perceptual assessment of color and how that may be modeled. So I realized when I was putting this together that there is a lot of stuff that one can talk about. So some of the material may sound a little bit out of your elements for you, so I'll try and introduce these as hopefully thoroughly as I can, but within the space of time that is available to me. And some of the other stuff may be more pertinent. So by the end of the presentation today, hopefully we'll have a better appreciation of what color perception and prediction is. So to start with, I know people run out of time in acknowledging help from others, so I decided to put this right at the beginning because a lot of people have contributed to the development of models, the experimental work, as well as generally speaking to the entire process, and least of all, of course, the funding, if you like, people. But I would also like to acknowledge the opportunity Dr. Trussell and the ECE has given me to come and talk to you about color. So with that out of the way, the people that are listed, Dr. Hinks is a professor in the College of Textiles. Lena Cardenas is one of our postdocs. Reed Clance, Single Lee students. Ralph Cooney, an adjunct professor. Roger Woodard is a statistician in the PAMS, and I have used stuff from NEAT's laboratory and some other people. So what are the objectives of this presentation? So very briefly, I'm going to review some of the color difference models that are used industrially. And most of these models use perceptual data. So hopefully we'll have an understanding of why these models don't perform the way they should. Okay? And then we'll talk a little bit about human color vision system, the variability amongst humans, which is very large, and some of the key variables that influence our perceptual assessment of color. Then hopefully I will have time, because we've done a lot of work over the past few years on various projects, and I will report on some of the important ones in terms of inter and inter-observer variability, and also examination of the role of observers' age, number, and experience in the assessment of color. So to start things off, one may ask the question why we need colors in the first place. Well, you can think about detection of an object. Do we require color? Probably not. But can we perceive information in a much more accelerated manner by looking at color? Yes. So the next image, from a survival from an evolutionary perspective, if you are a primate and you're looking for food, looking at a black and white image of the world may not really give you a lot of clues as to where to find these, and of course making that colored makes all the difference. So you can easily try and basically survive, hopefully, with the addition of color vision. So what about industrial applications? In terms of industrial applications, we need color because it's an integral component of many of the products that we produce. And in terms of quality control, one needs to be able to come up with models that will enable you to differentiate the difference between two similar colors. And also, can you reproduce the exact same color? How can you actually settle the issue? So there are visual processes for the assessment of either color difference or color resemblance between objects. And there are also colorimetric models. And the colorimetric models rely heavily on the visual data that are embedded inside them. So in devices like spectrophotometers, etc., we need to come up with a mathematical representation of perceptual data. One would think that there is a standard organization that deals with all of this stuff, and that makes it relatively easy to come up with a procedure to define color difference between objects. And like many other processes, one learns very quickly that there are indeed many organizations. In the case of color, International Standardization Organization is one of them. CIE, which is the main body responsible for work on illumination and color, is the other. And then in the textile industry, you have ASTM, which is the Association for Machinery Manufacturers. You also have Society for Automotive Engineers. And we have the American Association of Textile Chemists and Colorists. And these are all standardization bodies, so they each have their own procedure for the assessment of color, and sometimes these don't agree with one another. Okay, so one needs to come up with a way of communicating color without upsetting people in various industries. And one of the most desired, sought-after ways of dealing with color difference is coming up with a single number which one can use to identify the difference between two patches of color that may be different in terms of their hue, chroma, and lightness. So we are trying to represent a three-dimensional color using a single number. And the easiest model assumes that you have a uniform color space, a Euclidean space, therefore, a Euclidean distance, therefore, would define that color difference between the objects. But one quickly finds out that the system is not quite perceptually uniform. And a number of models and attempts have been made over the last 40, 50 years to generate other color difference models that perform better. So this is the CIE DE 2000 equation on the screen that was proposed in 2001. And you see a number of fudge factors that are introduced to take care of some of the issues, perceptual issues in the assessment of color, to make the system more uniform. So in terms of visual assessment of color, again, there are a number of methodologies. One can look at sample from different points of geometry of illumination and viewing. 45 degrees, you're looking down at an object or looking straight at an object. What type of illumination are you using? And these all influence your perception of color. So when you are dealing with color differences, and this is far from good, this image here, but the central patch is your standard, and the patches around it are the tolerances that are allowed in industry for making that color slightly lighter or darker, slightly redder or bluer, or slightly greener and redder, if you like, bluer, yellower, redder, and greener, yet still being acceptable. And you can see that the patches all look very, very similar. So industrial tolerances are very small, right? So you'd expect that the visual tolerances would also be kind of small. But hopefully you'll find that the visual tolerances are all over the place. Okay, so how do we actually define this? There are tolerance volumes, and you can actually define in relation to the center color shown in the object in the image there to your left, how much chromatic variation is allowed by S upon C, how much hue variation is allowed by S upon H, and how much lightness variation is allowed. And that shape is an ellipsoid. And again, that ellipsoid is not uniform in its shape, size, orientation across the color space. So when we are trying to model this variation, it doesn't come easily. It's not a trivial task. So as I said, there are a number of models from 1976's C-Lab and C-Love that is used in imaging more, all the way to the E2000 that was proposed in 2001. And they use various visual data for enhancing their performance. So you can see just right off the bat, looking at this slide, hopefully you will appreciate that each of the major visual data sets use different number of observers, different surrounds, different textures, and different assessment methodologies. And as a result, you'd expect that there would be some variation between these data sets. And the most recent one is the one that we produced. And this is just one color center, a lot of samples, and a lot of assessments, over 3,100 assessments for that. But this slide shows you the performance of some of the most important color difference equations. So from C-Lab to CIE94, CMC, and CIED 2000. And the axis here is PF3, which is a composite statistical model. Color scientists, some don't like it, but a lot of people have reported this as a means of correlating visual data to predicted data. So to give you an idea, if PF3 is 0, that means you have perfect agreement. Okay? The value that is reported here exceeds, in some cases, 50, 60. So roughly speaking, that shouldn't be percentile, but roughly speaking, if you think about it in terms of percentages, this tells you that color difference equations agree in the range of 50 to 70% with visual data. So it is from toss of a coin to slightly increased representation of visual data. So we are interested in finding out why there isn't sufficient agreement between perceptual data and predicted data. Okay? So some of the issues are related to the fact that mathematical models use a set of functions known as standard observer, and I'll talk a little bit about that. And the other one is due to human factors. How many people are being used that are tired when they are doing their assessments? Okay, how many times have you done this? What is the age distribution of the observers participating in the study? What type of illumination was used for the illumination of samples? Was it fluorescent? Was it calibrated tungsten? And so on and so forth. Then surround variations and variability in samples in terms of texture, the size of the sample that you're looking at, and some other factors. So mathematical models, everybody should know this. Of course, you have three sets of functions, starting with the spectral power distribution of an illuminant. You can physically obtain that using radiometry. Reflectance curves or emission curves, again, you can obtain that relatively easily. And the third set is the so-called observer functions. These represent your broadband sensors, human broadband sensors. So for red, for green, and for blue, they are averages. And you'll come to see when I talk about averages, it makes a lot of difference. So in imaging, people deal with small objects, small samples, pixels. So you are probably interested in a small field of view. In some other industries like paints, textiles, we are dealing with larger patches because we are interested in uniformity. So uniformity is usually dealing with 10-degree standard observer, which is the larger patch that you're seeing in relation to the smaller patch. So that is a relative difference. So standard spectral sensitivity functions that are now defined as a linear transformation of experimental data is really a statistical measure. And one needs to understand that it is influenced by the illumination conditions. So if you are using high illumination conditions, we are only employing our cones. If you use low illumination conditions, we are starting to use our rods. And if there's something in between, you may have the so-called rod intrusion. So some of the models don't take into account the rod intrusion that actually creates some problems. So again, from a physiological perspective, the highest resolving power in our eyes is around the small region in the retina known as fovea or foveola. And if you move further away from that region, the resolution significantly decreases, okay? So the 2-degree observer is very accurate, highly accurate, but it doesn't contain all the information that the observer perceives when one is looking at a larger image. So that's kind of important to bear in mind. So what is the standard observer function? So back in 1920s and 1930s, work was done in the UK, most famous classic works by 2 individuals, WD Wright and the J. Guild. And one of these guys used 10 observers, the other one used 7 observers. From the 7 observer study, I know that 1 was female. From the other one, we don't know the composition of observers and their age distribution, and that information sadly is lost. So what you see here is a range of responses from these observers that took part in an experiment that asked them to match colors using primary lights. And you see that there are differences between individual responses, and we don't use those individually. We use their average. In 1959, Stiles and Birch did another study. They used a larger number of people. They looked at the 10-degree observer, and they used 54 observers and then discarded responses from 5 observers that were considered erratic in their responses, okay? Yes, sir?
How is data like this generated?
Basically, you have a set of primary lights. You put an observer in front of a screen, and you ask them to match a color using a known quantity of each of these primaries, okay? So they look at red, and they adjust the amount of red primary and green primary and blue primary, and then you get a set of responses for each of the colors over the entire spectrum, okay? And then you average it, and you come up with response curves for an individual observer.
You might actually indicate a particular color takes this much red, this much green, this much blue on the graph there.
Right. I mean, I really don't have enough time, but I can tell you more later, okay, because I'm going to run out of time. What I am interested in here is the variability between observers, and you can see that between the least and the most variation, there is a significant gap. So how do we actually take into account that significant variation? Well, we use an average, okay? So what you have in instruments and models is 2 sets of average functions, one for the 2-degree, which is used in the imaging industry, and the other one for 10-degree, which is used in a lot of other industries. And you see that there is a difference between 2-degree and 10-degree. You'd expect that, and this is accounted for in models and in spectrophotometers, etc. But what is not accounted for is the large variability that you saw between observers, okay? So bear that in mind. Now, you would expect that using a standard observer and using all sorts of other mathematical models, you could come up with a uniform, perceptually uniform color space. Well, a lot of work has been done in that area, and this is the best so far. If you like, you've got the C-lab space, and you have here chroma contours and also equi-hue lines. And if this was to be a perfect uniform system, you'd expect the lines to be straight lines, and you'd expect the contours to be circles, right? So this is not the case. You see that in the yellow region, for instance, you have compression. And this is one of the problems in the modeling of color across the color space, because responses are not uniform across the color space, okay? So again, bear in mind that we are dealing with models that include statistical entities. We also are dealing with non-uniform representations of our perception. So how does our visual system come into play when we are dealing with color? So there is a lot of info in here. I'll go through some of that at a really fast pace because we don't have enough time to cover it. So when we talk about visual system, we need to differentiate between sensation and perception, okay? So with sensation, you can actually sense some kind of physical, if you like, a stimuli that hits your eye, and that physical stimuli may be converted to some kind of neurological response that is transmitted to your brain, okay? So what happens in your brain is the conscious decision at that point that uses that information to come up with some kind of response. And perception is not a fully understood phenomenon because we really don't know much about how brain works. But we are making big strides in terms of understanding the color vision part. So again, from a sensory point of view, we are interested in retina, of course, because that is where our detectors are. And the retina has a range of wires attached to it. Basically, you can go through a range of different types of cells that encode information. Some of these pass on information in a bipolar manner. Some do some other stuff. But basically, the information is transmitted to the visual cortex. And in the visual cortex, you come up with an experience of color. Okay? So is that experience similar for all people? No. Well, there are individuals, it's a known fact, there are individuals that suffer from what we call color deficiency, and there are different categories of color deficient people. You can have trichromats that behave slightly differently compared to your average trichromats, and they use different amounts of either red or green or blue to arrive at matches for certain colors. And then we have dichromats, and this is also very common in the animal kingdom. A lot of animals are dichromats, like your dogs, for instance. It is a misconception that dogs are colorblind. They're actually dichromats. Okay? So some of these guys behave as though one of their sensors is missing, okay, when they are looking at the color patches. And I'll show you some examples of that. And then we have monochromats, where individual behaves as if there is no color, if you like, sensor in their visual system. So for a dichromat, the mosaic of the retina may be represented by the two patches that I am showing you here. One of them shows you a protonope, which lacks the L cone, and the other one shows you a deuteronope, which lacks the M cone. And L represents the red, if you like, the long range, and M represents the middle range. And visual function, if you like, relative sensitivity curves are also shown there for your information. So how would the world look like to a color deficient person who is a dichromat and is missing one of the cones? The question is, which one of these pieces of meat is the most cooked one? Number 1, number 2, number 3, or number 4? And this is taken from Professor Neat's lab. If you have time, just search for his name, and you can Google his name and go and find some really neat images in his website. Can anybody guess which one is the most cooked meat here? 1, 2, 3, or 4?
3.
3? Yeah? Anybody else? All right, let's look. So number 4 is the most cooked, okay? And number 1, 2, and 3, in one case, actually, the monitor is not very calibrated here. So
You have to look at it on the
right, so you don't quite see it that well. But number 4 is the cooked one. So there are problems, of course, from a perceptual point of view. There have been really interesting studies that have happened recently with the so-called gene therapy to improve the problems associated with dichromats and some other animals. And here we have a study reported by Neitz and his co-workers. This is a squirrel monkey that was color deficient, and the world to this squirrel monkey would look like the image to your left initially, and there was post-surgery imaging taken on afterwards, and I have a video here which I'd like to show. It is on YouTube, so you can actually see what's going on here. So what this monkey is doing, after post-surgery, they show the monkey images of red patches, and when the monkey identifies the red patch, it gets some food. So you can see that the surgery has been successful in this case. The monkey has actually acquired the sensation that it was missing. Okay? So this is really very interesting work that has been done. And the question, obvious question is, can this be done in humans? And there are, of course, all sorts of ramifications for a study of this nature in humans from a psychological perspective and also from side effects, etc. But they are trying to obtain the required, if you like, guidelines from various industries to start doing some work in that area. Okay. What about other individuals? Are we all trichromats? Well, some genetic study has been done that shows the variation between genes that are responsible for this broad band, if you like, filters in our sensors is very, very small. Okay. So, for instance, the amino acids that are involved in a spectral tuning between S and L. So this is the short and the long wavelength. We are dealing with only 15 amino acids, so it's a really small number. Okay, with M and L, that is even smaller. And what happens is that there are theories talking about the evolutionary perspective of color vision, saying that really M is evolutionarily part of L. So these are polymorphic, and they are not very stable. So there are lots of people around that have different types of L's, for instance, and different types of M's. And you'll appreciate this hopefully a bit more in about a second. But even amongst others who have the same types of cells, the number of these broadband, if you like, cells is not the same. The ratio is actually heavily in favor of red, then to green, then to blue. And there are theories as to why that may be, because we don't get a good image with the blue in our retina, so we don't need as much of those and all of that stuff, okay? But the important issue here that I wanted to point out to you is that because of this polymorphism, there are some females around that have a fourth photoreceptor with a variation of about 12 nanometer. That makes it sufficiently large to make that individual tetrachromatic, okay? So that difference would be it's a huge variation in terms of the amount of colors that can be added to that observer. So if somebody told you that they are a super vision kind of female, don't doubt them, don't question them immediately, okay? So again, amongst other individuals, this shows you variations amongst color normal individuals, okay? And what you see here is the difference between L to M to S, if you like, cone ratios, changing from 1 for L to M, 1 to 2.7, all the way to 16.5 to 1. Okay? Now, you'd expect these individuals to report different sensations. And what is really striking with this study, I have the references for it that you can go and look up, is that they showed monochromatic light to these observers, and they asked them to describe their sensations of color. And the monochromatic lights were used in the 1 degree field of view. And in some cases, they used lights that had no S-cone, would have no S-cone input in them. Yet the observer reported that they were seeing or they were perceiving blues or purples. Okay? So what does that imply? Well, it has significant ramifications, because it implies basically that photoreceptors within the same class can elicit more than one color percept, okay, more color sensation. And from a theoretical perspective to model this, it's a nightmare, okay? So how can we actually come up with, with models that describe all of this variability? So visual cortex, again, a complex body of mass. We have interesting studies that have focused on areas in the visual cortex known as globs. And these globs have basically color coding of green or red and blue. And a recent study showed that they are even coding yellow. And the significance of this will be evident to you when I talk about the opponent color vision system in about a few minutes. Okay? So perceptually, even if we take care of all of that stuff, our eyes are not always providing us with the information that we can easily understand. here you have a range of gray samples with 3 different backgrounds, and you can see that by changing the background alone, you can create the perception of increased or reduced difference between adjacent patches. Okay, why is that important? This is, I wonder if we can get this working. All right, so this is a nice demonstration. So you can all stare at the dot in the center of this image for the next maybe 10-15 seconds. And this is a demonstration of how our brain fills in the information that sometimes we don't have. So you have an image, the next image, when you're looking at it, look at the same dot, same position. Don't divert your eyes to look at anything else. And tell me what you saw.
Oh.
Okay? So this is a black and white image, but you perceived full color sensation. Okay? So where did that come from? So the problem is, of course, our brain is filling in all of that information, and we don't really have good models that can explain this. Simultaneous contrast. Again, if you're looking at the 2 patches in the middle of the blue and gray, blue and yellow, are they the same or are they different? And they may appear different, but I can assure you by masking the background that they are indeed identical. Okay? And this is known as simultaneous contrast. And you can do the same with a larger, if you like, image. So would you say that the patch here and the patch here are the same, or are they different?
They look different.
It's different.
They appear different, right? Let's test that. Let's again put a mask on top of the cube, and you can hopefully appreciate that this creates all sorts of challenges for the modeling of color. How can we actually develop a model that can be mathematically accurate? Okay, so it is not a trivial task. So Herring proposed a model for the for the vision, color vision, and he proposed that we have opponent channels, and these opponent channels transfer light dark information as well as red green and yellow blue. And I showed you an image of a cortex, and I told you that there are studies that show there are cells in our brains that actually are responsible for the detection of these color-coded information. So this is new development. Okay, it hasn't been proven, but it looks like there may be neurological support for Herring's idea of this opponent color vision. So in this system, you're saying you have a center surround basically working, and if the center works, this is plus m minus l, you will see something in the green region, and if there's the opposite, it will go in the red region. And if you have plus l plus m minus s, you would see yellow, and the opposite would go for blue. Okay, so with this information, we can try and hopefully come up with better models if neurological model representation is found to support it, to increase the accuracy of our color mathematical models. So with this in mind, what have we done at NC State to improve the science of color? We have done quite a lot of work, of course, in different areas, but in the College of Textiles, we have looked at the so-called unique hue variation amongst observers. This is Herring's idea of opponent color system, right? So we tested a large number of observers, over 100, and we asked them to look at a range of samples with the full hue circle, and then tell us which sample represented the pure yellow to them, a yellow that contained no other, if you like, impurity in it. And the same for red and blue and green. And the representation of results on this Munsell hue circle is kind of striking. Just by looking at this, could you tell me what the problem is? You have a range of pure reds, a range of pure yellows, and the same for blues and greens amongst observers. So which one is the pure? Okay? Again, the perception varies amongst observers. And why is this important? There are color appearance models, And in color appearance models, one of the things that these models do is predict what hue it is that you are trying to kind of look at, all right? So to do that, they have used responses from a world study using natural color system values. And you have two ranges of what is the value that's currently used in the system, and what did we find in our study? What are the differences? So you can see that in the case of, for instance, yellow and blue, what we found and what the NCS has reported, that there is significant difference between them. Okay? So what does that mean for the accuracy of color appearance models? Again, which one is accurate? We don't know. All right? But we can tell that there are large variations between these systems. What about aging? So if you use a range of observers that are in their 20s and 30s, and then another range of observers in their 60s and 70s, which one is the more accurate one, or is there a difference between them? So the image, again, to your left shows you two pairs of lenses, one for a 79-year-old on top and the other one for a 39-year-old below it. And you can see that one is significantly yellower than the other. And you would hopefully appreciate that this yellowness would contribute to the perception of certain colors, for instance, blues. Okay, so if the blue is being filtered out and you're responsible for color matching blue samples, what is it that you're matching? Okay, so this, in addition to the fact that our accommodation power significantly decreases with age, raises a number of questions. You know, so what age should be the average observer that is used for visual studies or for perceptual studies? Certainly, if you are going just over my age, I'm at the age where my accommodation ability is nearly getting to be zero. Okay, so you can see that, you know, if you are dealing with younger observers, there may be better chances for good responses. But is that necessarily true? So we looked at a large number of observers, young, both male and female. We obtained a pair of lenses that mimic or artificially age that observer. So they put these goggles on when they were looking at colored patches, and then they took them off and they looked at the same patches again after, of course, a rest period. And they repeated this study 3 times to ensure that it was repeatable. And we looked at the responses. So you have the average male aged and average male responses, and also average female and female aged responses. And you can see that the differences in this case for females tends to be larger than for males. But regardless, for both population groups, the difference is statistically significant. Okay, so that means that the variation between observer groups has to be taken into account seriously. What about inter and inter-observer variability? So how many observers can you select and then still stay within the framework of having a set large enough so that it can be used in visual studies? So we've done a lot of work. We've used experienced observers versus naive observers. We call them experts versus naive. We also looked at, as I said, the effect of different types of assessment procedures. And we did an international study where we took our samples and the illumination box, the entire setup, and moved from country to country, employed 25 observers in each setup, and asked them to do this assessment for us, and then analyzed the responses. So I'm going to show you some things very, very quickly. This shows you a fishbone diagram that we developed that shows the key variables that influence the perception of color. Of course, it is a simplified fishbone diagram. I could use any of these arms and expand it to a full fishbone diagram. And this just shows you how difficult and challenging it is to come up with a model that accounts for all these variables. So I told you about our international study. One of the things that happens is that a given observer, if you ask them to repeat their assessment on a number of times, they will vary within themselves. Okay? And this variation can be large, can be small. So the image to your left on top shows you the variation within the same observer, okay, between trials. And you can see that it can be small in some cases. So this is a highly consistent observer, and very large in other cases, a very inconsistent observer. Okay? And then we have 3 patches of colors with different color difference values from 0.95, very small, to 2.97, and then to 11.7. And each of these lines is an average of 25 observers. Okay? So you have 100 observers in each plot. And what you can see immediately is that there are large variations between groups of observers. Okay? So intra-group variability is very significant. So we've looked at people from South America, people from North America, people in Europe, and also in China. And what we found out is that variability for observers in each trial, again, varies. But for the most part, the average, the mean, is about the same. Okay, but the variability is significantly large. So, all right. So what about the difference between observers that are experienced and those that are not? So we had a study where observers would sit in front of a standard calibrated viewing booth with a set of samples, assess the difference between the patch, the colored patch, using grayscales, okay? And we had a large number of people doing over 3,000 assessments, and we compared the responses from naive people versus experts, okay? And what we found out was that they track each other's responses very well. But for the most part, expert observers give you a higher bias, probably due to their professional, if you like, experience. Okay, so some people who are working in color industries are trying to show that they are highly discerning judges of color difference. Okay, so there may be some bias there, but this is statistically significant. Again, the differences are statistically significant. So I showed you in a previous slide a gray scale that is used for the assessment of color difference. That gray scale is geometric in nature. By that I mean the difference between patches increases roughly geometrically from 1 to 2. So we thought that may be contributing to this variability, and we tested that hypothesis by generating a perceptually linear grayscale. So the difference between this patch and this patch is meant to be identical to this patch and this patch, okay? And we tested our observers, again, looking at the same set of samples, and looked at a range of different types of assessment methods. But what's striking is that when we used our perceptually linear grayscale, we found that the variability was significantly decreased, okay? So that may be a contributing factor. So you can see this better here. Again, the range, the response range is within the same area, but the variability that you see is significantly reduced when we are using the perceptually linear grayscale. Another question that's a very important one: how many observers should one use when they're dealing with perceptual assessment? Should it be 1? Should it be 100? I was in a conference, and a prominent scientist told me, well, if you are a color normal observer, that's all you need, 1 person, okay? And they're right to a certain extent. But if you are dealing with the generation of visual data sets, one needs to understand there will be significant variability between observers, okay? So in different types of studies, we looked at 300 observers, looking at a range of samples. And this is one of those studies showing you 100 observations and the standard deviation that is obtained as a result of the number of observers employed in the study. And you can see that by increasing the number from, say, 2, 3, 4 to 10, you're still getting a lot of variability. And when you come to around maybe 25, 30, they're starting to plateau. So at the very least, one should use about 25 observers when they're dealing with visual assessments. This is our recommendation. So to conclude, I have made you run through a lot of different topics at a very fast pace, and I appreciate that it has been, in some cases, outside of your elements. But let me just conclude that color difference models have been improved over the last 30, 40 years. We have moved from a model that was performing at 50% agreement with visual data to a model that is performing at about 70%. So that 20% increase is very significant, okay? And but the problem is that change, for instance, from 50 to 70 is significant, from 70 to the previous model that was doing 67 is not significant. So more and more models are coming out, and of course to change these one needs to justify why do I need to change all the software, all the models that are employing this certain model in favor of the other, and has there been sufficient improvement to account for that type of change. And our argument is that when we tested different models, we found insufficient improvement from CIED 2000 to the previous model CMC, and therefore we did not recommend the industry to change the model from CMC to CIED 2000, despite the fact that there is some improvement, but not enough, large enough. And now there is a lot of work that's being done in color appearance modeling to look at the bigger picture. One does not look at a patch of color all the time. One looks at an image, an experience. Okay, so color appearance models are meant to account for the role of surround and a number of other things. And these are probably the way to go when it comes to modeling. We found out, hopefully, that human visual system is a very complex system, and we don't really know much about the formation of an experience in our brain at this point. There is no neurophysiological model that can explain all the things that we are actually perceiving and experiencing, but some models are slightly functioning better than others. So I didn't have time to go into different types of models there. We talked about inter and inter-observer variability, and I hope I have shown you that this variability is really large, even if you control the settings. If you use the same samples, same illuminations, you still get large variability amongst observers. I showed you that the difference between experienced and inexperienced observers is significant statistically. I also showed you that the difference between young and slightly aged observers is also significant statistically. So we talked about the fact that the use of a perceptually linear grayscale reduces variability in visual assessment of samples, and that if one is interested in the assessment of color on a perceptual basis, one needs to use a large number of observers. Okay, so at least 25, but in some studies probably more than 40, 45. So with that in mind, I thank you for your attention, and I'd be happy to respond to any questions that you may have. Yes, sir.
So what kind of real-world applications would a model like this be useful, and at what point, I mean, how good is good enough for a particular application?
Right. That is the question, really. You know, how good is good enough? And we are trying to come up with models. Models are used in every industry. In imaging, when you're looking at a color on a monitor, and that color is to be produced on a piece of paper, and then, of course, reproduced on a piece of fabric, they need to match. Okay? So the color differences that are calculated between the monitor color and the fabric and the paper, they have to account for some of this variability, because all of those models, as I told you, at their heart, they employ this perceptual data that are gathered from different studies in different parts of the world. And our argument is that one needs to account for variability amongst observers and improve that core data to kind of make the model perform better. But how good is good? Well, right now we have with CIED 2000 about 70% agreement between the two different systems, visual versus predictive. Okay, and many people use it. In the imaging industry, the 2000 is now recommended by the CIE. As I said, in the paint industry and textiles, CMC is the recommended right now by various standardization bodies. But obviously, we are trying to improve it further. Now, one of the issues that we have been very careful about is that probably we won't get to a point where there is a model that agrees 100%. That is going to be impossible. But if we can improve it, say, to about 80, 85%, that would be slightly better than what it is right now. So I think lookup tables may be one way of doing this. Coming up with other color appearance models may be another. And you know, trying to improve the mathematics of the color space so that it becomes more uniform may be another. Yeah.
I had another question. Were there any significant results of the international
Replication
experiments?
study? Yeah. In what way? Significant?
Do certain parts of the world perceive more accurately than others, or in different ways that are significant?
That's a good question. Actually, in the study that our PhD student did, she found that the observers in Colombia were the most inconsistent, okay? But that may be a biased statement, because most of the observers in Colombia were 17 to 22-year-olds, okay? Whereas when we did this study in Czech Republic as a good representation of Europe, the average age was about 27, okay? And they were, again, observers in that group that were highly inconsistent. But for the most part, you know, we found that, I think I showed you in the last slide that, for instance, our Chinese observers tend to be more consistent and kind of more accurate in their assessments. So accurate is a rough term I'm using here, because every person is accurate, obviously. It is difficult to say with the limited number of people that we studied that there are genetics in play that make a group of people more discerning of color differences than others. Yeah.
In comparing an image or something that we're trying to detect, then the surround, you demonstrated on several occasions, has everything to do with it. Now, but what about texture on a finer scale there? How does texture affect the perception of color? Mm-hmm. And then how is it actually measured?
That is a great question. Of course, with textiles, we are dealing with texture. Okay, so we found out through experimentation that if you add texture, it influences your perception of color difference. And the more textured an object is, the more significant the role of lightness becomes, for instance. Mm-hmm. So in CMC, color difference equation, and in the other equations that were proposed later on, there were scalars, functions that were included in the color difference model that account for the role of texture. For instance, CMC 1:1 presumes that the surface is flat when one is looking at it, and CMC 2:1 considers a textured surface. In the carpet industry, where you have large piles and light is going to get lost when it is traveling towards the object, the role of texture becomes even more significant. So there have been proposals for using 3:1 in the case of textiles, highly textured textiles. But we have actually an ongoing study looking at a range of textile samples dyed with the same amount of colorant, but the only difference being that they have different textures. And we have used, again, observers looking at the samples and defining their experience, and we found that texture, of course, is a significant player when it comes to perceiving color. So I don't know if that answers your question, but we are working to kind of account for the role of texture. It is an ongoing study, and it's an important one because, of course, if you are trying to mimic the image on a screen, how much of that texture is being reproduced on a screen, and is that providing you with the experience that you are otherwise getting when you're looking at the physical sample? Yeah. Yes, sir.
You said about the number of observers. You said you need just one observer if it's a color normal observer. Mm-hmm. So how would you determine what is a color normal observer?
Well, there are tests that we can carry out to detect if any deficiency that one may have. You know, there are so-called isochromatic confusion plates, and there are tests of Neat's lab, and other people have different types of tests. So anybody that comes to our color science lab to do any kind of perceptual study is first tested for color normalcy, okay? And once we are sure that they are color normal, then they carry on. Because we have individuals, amongst the male population, for instance, about 8% are color deficient. So it is big, okay? So we try to, of course, screen for color normalcy first. Yeah, and there are very easy methods of doing this.
in the world of imaging, people have tried to develop color enhancement methods there, and to a certain extent, you know, the people in the film industry and camera industry have biased things there. But what does the quantitative color measurement and the variability of observers say about the goal of color enhancement? Is this a possible goal?
It may be possible. When I attended a conference last year, people had come up with a new term, vividness, and one of the prominent scientists there kind of raised his hand and said, "At what point are we going to get rid of these nesses? You know, because we are talking about chromaticness, colorfulness, vividness, and you can go on and on, talking about different sensations that one presumably perceives when one is looking at color." But the response from the speaker was that, well, we are developing color screens for the TV industry, and people talk about vividness of color when they are looking at the images on the monitor. So we were interested in generating a model that represented vividness, for instance. So enhancing color, what does that actually mean? You know, one can try and enhance color from various perspectives. Obviously, one can try to increase the gamut to kind of make that more saturated or more chromatic, etc. But there may be systems out there. Our ultimate goal is to try and come up with mathematical models that generate sufficiently accurate representations of human experience. And so how can you do it without really getting into the nasty science of statistics? And I use that in a very crude way. But statistically, one can argue a lot of different things, and one can use the data to represent different stuff. But I think there are models out there that can do this, enhance our color experience, but how accurate they are, it's open to questions.
Okay.
All right.
Let's thank our speaker.
Thank you.